Hello, everyone. Good afternoon. My name is Maya Corin, and I am an assistant professor in the Department of Environmental Medicine and Public Health and a member of the Mount Sinai Institute for Exposomic Research. It's an absolute pleasure to welcome you all to the very last session of the 2023 Lunchtime Chats, where we will discuss the fascinating relationship between metal exposures during early life and their impact on brain development. My work focuses on how to best communicate information, health information in particular, to the public. So I love how these lunchtime chats provide a bridge between the important science taking place here at Mount Sinai and how the public can make use of this information. Today's talk is about metals. Metals are one of the most studied compounds in environmental health because of how common they are in our environment. They are found in air, water, food, and all the different products that we use every day. They can also impact our health in various different ways, including how the brain develops and functions. Metals such as mercury, lead, and arsenic have long been known as neurotoxins and can have really profound effects on the developing brain, particularly in early life. At Mount Sinai, in the field of exposomics, we're studying environmental exposures across the lifespan and understanding how the windows of vulnerability are key. The timing and duration of these exposures can really have a significant impact on health outcomes, particularly during these really critical periods of development. Prenatal and early life exposures to metals are of particular interest as the developing brain is highly susceptible to the toxic effects of metals, and exposure during these critical periods can lead to long-term neurological deficits. Our wonderful speaker today, Dr. Megan Horton, will lead us through this maze of knowledge. She is an associate professor at the Department of Environmental Medicine and Public Health at Mount Sinai and is also a member of the Institute for Exposomic Research. Her research combines state-of-the-art environmental exposure assessment with structural and functional neuroimaging and behavioral phenotyping to understand how early life exposure to developmental neurotoxins impact typical brain development and leads to aberrant cognitive and behavioral outcomes in children. Her research also investigates how environmental, social, and occupational stressors impact later life health outcomes, including PTSD and cognitive impairment. The presentation today will be about 25 minutes, followed by 20 minutes for audience questions. Please submit your questions using the Q&A functions, and if we can't get to them all, we will work to get answers shared on social media. Dr. Horton, please take the stage.
Thank you so much, Maya, for that introduction, and thank you to the Institute for Exposomic Research for the opportunity to present what we're working on today. So let me share my screen. Good. Great. Thank you again. Today, I'll talk about early life metal exposures and brain development, and mostly what I'm focusing on is some new tools that we've developed in my lab and in collaboration with other researchers at Mount Sinai that are allowing us to ask new questions about metal exposure and exposomic exposures and brain development throughout childhood and adolescence. Many of you have been participating in this seminar series for the last couple of years, so are well versed in the concept of the exposome, but I would be remiss for those of you who are just joining to not remind us what the exposome is and how we define it. The exposome has been defined as the measure of all exposures of an individual in its lifetime and how exposures relate to health throughout the life course. The concept of the exposome was developed to draw attention to the critical need for more complete environmental exposure assessment in epidemiologic studies. Environment in this context is defined as everything that's non-genetic and complements the genome by providing a comprehensive description of lifelong exposure history. It includes both adverse risk factors such as smoking, toxicant exposures, socioeconomic disparities, as well as positive or protective factors, including sleep, green space, adequate nutrition, paternal or parental nurturing, etc. So the goal in epidemiology is to understand how these environmental exposures can cause adverse health outcomes and how we can leverage this knowledge to prevent human disease. My group is very interested in particular parts of the exposome. The exposome is a pretty broad concept. I'm very interested in exposures that are occurring early in life right around birth in these critical periods of brain development occurring from conception throughout early childhood, and how these exposures early on could impact the trajectory of brain health throughout the life course. So today I'll focus on 3 specific concepts and tools that we're using to disentangle these relationships. One, we'll talk about the concept of critical windows. What's happening in the brain very early in life that could impact what happens later in life. So this concept of critical windows of development. We'll talk about exposure to mixtures. We know that individuals are not exposed to a single chemical at a time, but rather a chemical soup that contains both adverse or toxicant chemicals as well as protective chemicals. How do we start to understand what the effects of those mixtures are on a health outcome? And what do we use to look at brain health? So I'm interested not just in behavioral outcomes, but in understanding the mechanisms that impact those behaviors. So what's happening in the brain? So I'll talk about what tools we use to look at brain development. So to address these tools, in order to understand critical windows of development very early on, and to understand how chemicals very early in pregnancy and early childhood can impact it, we leverage the technology developed by Manish Arora and Christine Austin here at Mount Sinai that allow us to use naturally shed deciduous teeth or baby teeth to objectively reconstruct prenatal exposures. I'll talk next about the statistical methods that we use to manage this data. This is very high dimensional exposomic data. It's no longer looking at a single chemical at a single time point, so it requires more sophisticated statistical methods. So I'll talk about weighted quantile sum regression, which was also developed by investigators here at Mount Sinai. And finally, I'll talk about magnetic resonance imaging, or what we use to look at in vivo associations between prenatal exposures and early childhood exposures, and what the adolescent and childhood brain actually looks like. Why metals? As Maya mentioned, metal exposure is ubiquitous worldwide, and exposure in children has been associated with a wide range of adverse outcomes, ranging from intellectual disability, neurocognitive, behavioral and respiratory disorders, cancer, cardiovascular disease, etc. And despite legislation and consumer changes that have reduced exposure to several neurotoxicant metals, such as lead, cadmium, and arsenic, many children worldwide are still exposed to low levels and high levels of these metals. And it's interesting to note that the developing brain is more vulnerable to these toxicant exposures, particularly metals, than the adult brain. Children are not just little adults. They differ physiologically. Children eat, breathe, and drink more relative to their body mass than adults, and they have underdeveloped defense systems. Their brains are more permeable to chemicals crossing them. So chemicals that we're talking about, so many metals can cross both the placental barrier and the blood-brain barrier. Developing infants and children have less effective filtration in their nasal passages and their lungs. Their skin is more permeable, and their metabolic pathways and vital organ systems are still developing, so they may not be able to metabolize or clear many of those toxicant chemicals. Children also differ in their unique activity patterns and behavior. Children crawl and play close to the ground, putting them at risk to more contact with exposures from dirt and from dust. And as many of us parents know, children have increased hand-to-mouth activity. They put more things in their mouth, including their hands, so they could increase their risk of exposure. And then, as I talked about, there may be unique windows of susceptibility or critical windows of development that make these developing brains more vulnerable to adverse chemicals. So let's dig a little bit deeper into this concept of critical windows of development. Much of what we know about critical windows came from the thalidomide disaster of the 1950s. This idea that the placenta was a perfect barrier was current in the 1950s, when physicians believed that a toxicant would affect the fetus only if it caused major illness or killed the mother. Pregnant women were not counseled about the dangers of medications or alcohol. New drugs were not thoroughly tested during pregnancy. So nearly 60 years ago, thalidomide was described to treat morning sickness, or some of the adverse outcomes associated with pregnancy, like tiredness and fatigue. What followed was the largest man-made medical disaster ever, where over 10,000 children were born with a range of severe and debilitating malformations, such as these shown in this picture. So what we saw was that women who took thalidomide during their pregnancy had children who were born with cleft palates or this unique phocomelia in their arms and in their legs. Because we had medical records knowing when these women were prescribed and when they took this medication, we could look to see the strong associations between the stage of development where the infant or the fetus was and what the adverse outcome was. So you'll see in this graphic that we're looking at the days post-conception, and you can see if a pregnant woman took her thalidomide early in her pregnancy, you would see those adverse outcomes such as an external ear missing. However, if she took the drug later in her pregnancy, the external ear would be fine because it was fully formed, but there may be another host of problems depending on what organ system was developed at that time period. So this really led to our understanding of critical or vulnerable windows of development. So this is another slide just demonstrating that an organ that's undergoing development, such as in this highly sensitive period highlighted in purple, is more vulnerable to the adverse effects of a toxicant or of an adverse exposure. However, the less sensitive periods are when that organ system is fully formed. Then it's less vulnerable to the adverse outcomes. So the take-home point of the research that resulted from some of the thalidomide epidemic was that an exposure that's coincident with the developmental process is more likely to cause adverse effects. When we think about this in terms of the human brain, we have a very long and extended critical window. The brain begins developing immediately post-conception, and these graphs represent the timing of the development of three main domains in brain development: cognitive function, language development, and sensory pathways such as visual or hearing. So you can see this rapid increase in the slope of higher cognitive function beginning in pregnancy and extending throughout the first years of life. We now know that the brain continues developing. This is from adapted from a graph that was generated in the early in the century. And if we could redo this, which I'd like to, you would see that this line actually extends further out up here, because we know now that brain development isn't considered complete until very late in adolescence and early adulthood, so about 25 to 30 years old. So if we have this critical window very early in pregnancy, what kind of biomarkers should we collect so that we can identify exposures occurring during these periods? Traditional biomarkers of exposure during this perinatal period largely include maternal blood and urine, and occasionally cord blood samples. Some of the drawbacks, I guess I should start with some of the advantages of these, are that they're fairly easily collected. You can collect blood from a mom through venipuncture, and you can collect urine into a sterile urine cup. And cord blood can be collected from the cord at delivery. However, because we don't want to put too much burden on the research subject of the mom, we don't want her to come in too many times to give an extra blood sample. And so many times in research studies, these biomarkers are collected at just 1 or 2 time points. And then we try to make an assessment or an assumption that these exposures are continuous throughout pregnancy and early childhood. What we've learned is that these are indirect proxies for fetal exposure and may not reflect fetal or early infant exposure. We know that from an example with chlorpyrifos. Much of my research focuses on prenatal pesticide exposure. And interestingly, in a study at Columbia University that we did about 15 years ago, we measured chlorpyrifos, which was a residential pesticide, in both mom's blood around the time of delivery and at cord blood. And we found that the levels of chlorpyrifos were significantly higher in the cord blood samples than in the mom's blood samples. This is just support that the maternal blood sample may not be a good proxy for fetal exposure, because in the case of chlorpyrifos, the fetus was unable to clear that compound because its metabolic system was not online to be able to metabolize and clear it. Because of this deficit in understanding of prenatal exposures and early life exposures, Manish Arora and his team leveraged the physiological properties of teeth to start to measure, to objectively reconstruct early life exposures. So teeth grow very similar to tree rings, and we'll talk about this in the next slide, but they're also a fairly easy biological marker to collect. kids lose 20 deciduous teeth between the ages of 5 and 13. And because teeth begin growing in the second trimester of pregnancy and storing anything that was circulating through the fetal bloodstream, they can act sort of like a computer hard drive to allow us to reconstruct the exposures. Just a real quick diagram of what the tooth looks like. We're looking at the enamel, which is the part of the tooth that's erupting from the gum, the primary dentine, the secondary dentine, and then the pulp where the blood vessels are connecting the tooth with the fetal circulation. And when we look at how the tooth develops in this graphic, you can see that the tooth is layering layers of enamel and dentine on a daily basis based on the mom's physiological processes. And then the tooth starts to grow downward as the enamel erupts from the root or erupts from the gums. And critical to this work done by Manish and Christine is the development of the neonatal line at birth. So whether or not the mom delivers through a vaginal birth or a section, there's a neonatal line that's formed and visible in a confocal microscope that allows us to mark or anchor birth and then backtrack retrospectively to see how many of these layers were from the prenatal period and layers that are developing postnatally. So we can timestamp and look at each one of these layers. And you can see that with this confocal microscope and this neonatal line here, allowing us to use this as a timestamp. This is an example of lead levels in a tooth from a subject in Mexico that I'll talk about in a little bit. And what we're looking at here is the levels of lead. So this is an ICP mass spec, colored image showing low lead in blue and high lead in red. And these arrows relate right here to the timing of just about the third trimester. So we're looking at lead on the y-axis and time on the x-axis. And this is birth, denoting when the child was born. So these are prenatal levels and these are postnatal levels. And you can see that there's this spike in lead levels in the third trimester, which is consistent with what we would see in an infant who is born to a mom that may have lead exposure because of the skeletal remodeling and growth in the fetus in this time period in the third trimester. That fetus is pulling all of the natural nutrients, so calcium, out of the mom's skeleton, out of her blood, and mistakenly pulling lead if there's lead circulating as well. So we can do this now, not just for lead, but for many different metals, including zinc and manganese. And even this slide is slightly dated because when I started my work with Manish, we focused on 3 different metals. We now have more than 16 different metals that we can measure in the teeth. So we have more of a metallomics approach to looking at protective and risky chemical and high-risk chemicals and brain outcomes. So in order to examine the associations between this high dimensional metals data that we get throughout pregnancy and early childhood, we need to develop some advanced statistical methods. So we're no longer looking at just a single chemical and a single outcome. We're looking at a whole mixture of metals, and then we're looking at how that metal mixture changes over time. So we use a method called weighted quantile sum regression, which in brief is a method that allows us to look not just at the single chemical and the outcome, but that whole mixture of chemicals and how those affect the outcome. So in this diagram, I'm representing the brain because we can look at how this mixture of metals impacts the brain. And there's a couple very positive things about the WQS approach. One being that we can look at the effect of the overall mixture. And once we see that there's an association between the mixture and the outcome, we can backtrack and see which one of those chemicals, was it manganese, zinc, or lead, or some combination of them that had the most impact on this outcome. We can also look to see, were some of those chemicals protective against an adverse outcome? In the recent years, Chris and Paul at Mount Sinai have further developed this method to allow us to look not just at exposures that are occurring at a single time point, but to look at exposures that are occurring in weekly intervals throughout gestation and early childhood. So they expanded the WQS to include what is called a lag WQS. So this allows us to look at the moving mixture over time. So just a quick reminder that WQS is at a single time point. Lag WQS allows us to look at what's happening in week 1, week 2, week 3, all the way through pregnancy and early childhood, and how does that mixture impact the developing brain? And we can still go back and do the same that we did with the WQS, where we can identify what week was the most vulnerable, and within that week, what were the chemicals that were the most impactful. So I wanted to give you a preview of what this data will look like. And so when I show you real data, so we're looking at time since birth again. So with birth being 0, so this is prenatal and this is postnatal. And when we map the results of the WQS, what we want to see is whether there's a divergence from 0, where there might be an indication of a critical window or a sensitive window. So I'll show you some real data in a couple of slides where we look at this in a study in Mexico. So the final key to this is what are we doing? How are we looking at associations in the brain? How are we looking at associations with the developing brain? And for that, we use magnetic resonance imaging. So magnetic resonance imaging, or MRI, scans work as an imaging method due to the unique makeup of the human body. All of the cells of our human body contain water, which is principally made up of hydrogen atoms. The magnet that's embedded within the MRI scanner can act on these positively charged hydrogen atoms and cause them to spin. So if we're looking at our hydrogen atoms in our cells outside the body, they're randomly oriented or randomly precessing. Once we put them in a magnetic field, they're all going to line up in the same direction. By varying the strength and the direction of the magnetic field, we can change the direction of the spin, enabling us to build layers of detail. When the magnet is switched off, then these hydrogen atoms will precess or return to their original state. Fundamentally, the different types of tissue within the body return to their normal state at different rates, and that allows us to very finely, with good spatial resolution, visualize and differentiate between different tissues of the body. So importantly for brain imaging, we can see the difference between gray matter, white matter, and cerebrospinal fluid. In our research, we focus there's many different modalities. So you can look at structural connectivity. In our research, we leverage one modality of MRI called resting state functional MRI. In brief, functional MRI uses the properties of blood and oxygen to observe activated brain regions. So functional MRI is a proxy for brain activation. We're looking for where certain areas of the brain are activated. And this is based on this graphic up here, where we see in a basic state or in a non-activated state, the oxygenated blood and deoxygenated blood are circulating very evenly. However, when there's a demand on the brain, there's an increased blood flow to supply oxygen and glucose, and that oxygenated blood looks different in the MRI scanner than deoxygenated blood. So we can use that to look at activation states in the brain. Importantly, we can do this when the brain is also at rest. So this is looking at brain regions that may be correlated even when the brain is resting and not accomplishing a task. This is very useful when we're doing children's research, because often when you ask a child or an adult to do a task in the scanner, such as tap their fingers or focus on something, they start to move and wiggle, and movement and wiggling will disrupt our ability to get a good MRI scan. So when you ask somebody just to lay still, daydream, focus on a single mark in front of them, maybe a scenery or an X on the screen, and just ask them to space out. Even when they do that, we can see significant correlations between brain regions that we know are acting together at rest, like the left and right motor cortex, which is very different than areas of the brain that are not activated together, such as the left motor and left visual cortex, which are less correlated in this diagram. So how do we put this all together? We put this all together in the progress study in Mexico City, which is a study started by Dr. Wright and Dr. Tellez Rojo, our collaborator in Mexico, where they enrolled about 1,000 pregnant women between years 2006 and 2011. They performed extensive exposure assessment, including collection of the teeth. And they've followed the kids now, I should update this, now the kids are almost 16 years old, and they focus on respiratory outcomes, growth and obesity, and neurodevelopment. So several years ago, I started a study where we brought in a subset of these kids to do MRI. So ultimately, we could look at the association between prenatal tooth exposures, behavioral outcomes in the kids, and brain imaging outcomes, and see if we could tell a story about what was happening between the exposures, the brain, and the behaviors. This is an example of metal distributions in teeth. So as I said, we've now expanded well beyond those first 3 metals, and we're looking at multiple metals. So this is looking at the concentration of metals on the y-axis in 2 subjects that I pulled randomly from the progress study. And you, at this point, don't need to know exactly what those metals are, but that there does seem to be a lot of range in the distribution of these chemicals over that prenatal and early childhood period. If these lines were all flat, then we could use one single time point as the association with the outcome. But as these are moving a lot over these time periods, we'd like to know, is this peak here around 20 weeks critical? Does this have an effect on the outcome? And only using that lag WQS could we start to see that. So we use the BASC to look at behavioral outcomes. This is an assessment of behavioral and emotional outcomes in kids. And so this is, again, that map that I showed you before, where we're looking at time on the x-axis, or sorry, we're looking at weeks on the x-axis, and the association between that metal mixture and behavioral outcomes on the y-axis. And so we're looking at is, as metal mixtures are increasing over time, we do see an increase in behavioral outcomes. So this is in the positive direction, looking at that harmful association. And it seems to be that we have this postnatal critical window. It doesn't look like metal exposures in the prenatal period are strongly associated with behavioral outcomes. We're really seeing this association in the postnatal period. So what we did is look and see what metals in this postnatal period, particularly right here at the peak, which we saw at 43 weeks, what metals were most associated with this outcome of increased behavioral problems. And we saw that in our preliminary data, this is a small sample size as we're not done with the study, we saw associations between arsenic, manganese, and tin with our outcome. As I mentioned with WQS, we can look in the opposite direction. So we can look and see, were any metals in our mixture associated with a reduction in behavioral outcomes? And we did this again, and we saw that, interestingly, the same critical window, roughly, of that postnatal period, where we saw that increasing exposure to metals was associated with lower rates of behavioral outcomes. And the metals driving this association were different. We saw barium, lithium, and copper, which is interesting because many of these chemicals are not known as the paradigmatic neurotoxicants like manganese and arsenic. We next look at the association between this metal mixture and actual brain outcomes. So we used our resting state analysis that I mentioned before, and Azura and Renitsia, a postdoc in my lab, used graph theory resting state analysis to look at a metric called eigenvector centrality, or a measure of influence. So you look at different areas of the brain and look and see how associated or how connected are these areas of the brain with one another. And areas of the brain that have the most centrality or the most importance are considered important brain regions. And so what we looked at was, does metal exposure change those brain regions that have higher and lower centrality? Or does it shift a measure of connectivity between different brain regions? What we saw, again, you're familiar with this lag WQS map now, or figure, where we're looking at weeks on the x-axis and this time varying association on the y-axis. And interestingly, we do see a significant association between the metal mixture and reduced centrality in the insula, which is an area of the brain that may be associated with these behavioral outcomes. We saw that this association was driven by manganese, copper, lithium, and magnesium, so a mixture of both those protective and toxic chemicals. The results of this need to be disentangled a little bit. So what does this all mean? What does it mean that we're seeing an association between this mixture and behavioral outcomes and brain outcomes? Do we have enough information to talk about where we could intervene or what metals are intoxicants? At this preliminary stage, we're not able to say that. So we're powered to look at 250 subjects, and the preliminary data that I've presented to you is on about 80 subjects. So I believe that the results that I'm showing demonstrate the robustness of our methods, but the interpretation of this has yet to be determined. And once we have our final sample size, we'll be able to disentangle which may be the most protective chemicals and which may be the most toxic chemicals. But I think I have hopefully convinced you that the baby teeth allow us to reconstruct gestational exposures with high temporal resolution, that the lag WQS is an advanced statistical method that allows us to examine early life critical windows of exposure, and that magnetic resonance imaging allows a powerful and safe in vivo assessment of structural and functional brain changes that may be associated with early life, informing the mechanisms through which environmental chemicals may impact behavioral outcomes in kids. I think I'll stop there and ask for any questions, and then we can move into a little bit about what we do about metal exposures. Maya?
Yeah, okay. You hear me okay? Great. So my first question is around a little bit, if you can clarify a little bit about manganese. Is it both a protector and a neurotoxicant, and how does that work exactly?
It's a great question. And I find the chemicals such as manganese to be some of the most interesting to study, particularly with a method like WQS. So as opposed to something like lead, which the body has no physiological experience with, and there's no known physiological benefit. Any level of lead that we see is a neurotoxicant. So there's no positive benefit to lead exposure. Manganese, however, is physiological critical to many processes in the body, including brain development. So the body needs to have a physiological level in order to maintain development. If that level is either too low or too high, it can be considered a neurotoxicant. So if you have high manganese, it may disrupt the processes. And if you have manganese levels that are too low, it can also have adverse effects.
Okay, great. Can you actually stop your share so we can put up some of the resources?
Absolutely.
Great. Another question for you. Your study in Mexico, you mentioned that they had actually considerably higher air pollution exposure. And how do those exposures relate to those in the US and other countries?
It's another good question. When we started the study, or when Dr. Wright and Mara started the study almost 20 years ago, you could see from the picture that I showed in Mexico City in that time frame that levels of air pollution were considerably higher due to a lack of regulations. Lead was used in gasoline in Mexico City for considerably longer, up until several years ago, I think, while in the United States it's been regulated since the 70s. So some of the toxicants that we're seeing are higher in Mexico City than they are in the United States. However, due to continued exposure to lead-based products and other products that have heavy metals, when we look at comparisons, particularly in the teeth, between levels that have been absorbed by the children now and children in the United States, they are similar. So we might see subtle fluctuations in some particular chemicals, but there is still some similar exposures between the United States and Mexico.
That actually brings me to a question that I'm not sure how much you know about this, but I know that there's been a lot of work done at Sinai focused on 9/11 responders and people that were on the ground. Has there been any work around women who were pregnant and their young children who lived in the area and their exposure to some of these metals?
Yes, Columbia University has a, I believe it's the World Trade Center responder or family study. And during that time, I actually worked as a recruiter on that study when I first moved to New York. We enrolled moms who were exposed to 9/11 in the 9 months of any point in the 9 months of their pregnancy. And I know they followed those kids for at least 3 to 4 years and looked at associations between exposure and gestational age, and different metrics like head circumference and birth outcomes. And those results have been published. And I think that there's still a study from NYU that's following those kids throughout their teen and early adult years. So I don't have a good grasp on the literature, but it's definitely been published.
Oh, great. And it's crazy that they're now in their 20s, right?
Yeah.
Isn't that? Oh. Um, one other question was, how can people enroll in the tooth studies and have their baby teeth tested and analyzed? People apparently save their baby teeth.
I'm very thankful that people save their baby teeth. That's what we rely on for our studies. So I have to emphasize that, I mean, there's two different answers to this question. One, much of the research that we do is ecologic research. So I'm not looking at individual children. I'm looking at groups of children, and we're looking at small deviations from normal. So in the studies in Mexico, the studies in the United States, and I have a study in Italy, it's very unlikely that we would see an association between the metal exposures that we see in teeth and a diagnosis with a child. You may not even see that this child actually has a slight increase in behavioral outcomes on the BASC or the other scales that we administer. But what's important for us to look at in these research studies and ecologic studies is the slight shift of the distribution. So if we think about this, if we think about the distribution of IQ, right? We have an IQ with a median of 100, and we have a 15 points of a standard deviation below. And if you're under 85 points, then you're considered at some sort of delay. And if you're over 15, then you're considered gifted. What happens when we shift the curve just a few points is that we shift more kids into that delayed category and shift more kids out of the accelerated category. And while we might not see an individual difference, we see the difference in the groups. So when we're talking about some of these low levels environmental exposures, I don't necessarily suggest that it's the wisest to look at environmental exposures in the kids on an individual basis, unless there's some sort of risk factor that's known. If you're an artist and you work with a lot of metal shavings and your child was an infant crawling around, then it might be important for you to measure that as much as it's important for you to remediate that. But that being said, there are methods that are being developed to use those things like the teeth and the hair as markers that are associated with disease. But that's not what we do in research.
Right, understood. Can you talk a little bit about expanding the mixtures that you're looking at to include non-chemical exposures as well, like air pollution or stress?
Absolutely. I think that's a great question. We have a lot of information on these moms and the children throughout their lives. So we don't have just metals, but we have phthalates, we have flame retardants, other organic pesticides. And so we do work on trying to combine those step by step. So I really wanted to understand how we could start to look at the metals and in particular differentiate the protective metals from the neurotoxicant metals. And once we have a good grasp on that, then I think it would be great to pull in some of the other exposure metrics that we have to look at a broader exposomic approach. And we would be remiss if we didn't bring in some of the social factors, such as maternal and childhood stress, socioeconomic disparity, even things like discrimination and structural racism, and how those can combine and impact the effect of chemical toxicant exposures. And so we are starting to do that in some of our studies. I have a new PhD student who's very interested in combining metal exposures with stress exposures and looking at some of these brain outcomes into kids. So it's definitely a smart thing to do, and we're working on that.
Kind of bringing it with this kind of included panel. So the arsenic data that you have, arsenic seems to be so ubiquitous in many of the things that we are around us. Are the arsenic levels that you see, are they coming principally from food, or are they coming from other sources? Do we know exactly where the arsenic is coming from?
So in the study that we're doing in Mexico, we don't do source attribution when we're looking at the levels in the teeth or levels in the blood or urine. But we can look at associations between some of the questionnaire data that we're looking at and try to understand where are these chemicals coming from. With the arsenic, I think the general understanding is a lot of it is coming from contaminated rice and water. So one easy fix for not necessarily easy, but one logical step if you have concern about arsenic exposure is to have your water tested to see if you're living in an area that has arsenic, and should you use a filter to try to reduce your arsenic. And there's considerable literature. I think I have one slide, but I'm not sure if I have it, about trying to reduce arsenic and other heavy metal exposures in your baby food and your children's food. And there is, we can share that on the website if it's not already up there. But there are some suggestions even from our pediatric environmental health and safety unit, our PESU, or other, even EPA and CDC have some guidelines for trying to reduce heavy metals and arsenic in your baby food, which is one of the biggest sources of exposure in children.
That kind of brings me to the next question, which is, how do we educate policymakers to understand these health effects, right? So how do we kind of expand this knowledge into making real policy changes? And how do we work with the community as well?
I think that that's swinging the pendulum back to getting people to understand and believe in science is important. So I think working with kids and working with multiple generations, I found is very powerful. So getting, I think we have to use our whole base and not just rely on the policymakers, but to rely on the consumers. And the consumers are are learning things from their children, right? And from their communities. So it is incredibly important to work on legislation and work with policymakers and those types of leaders. It's also really important to work with community groups and consumers, because they can really push the envelope or push the needle in terms of legislation and I guess taking advantage of the market, and pushing what they will purchase and won't purchase based on how safe or clean it is. So
that brings me a little bit back to you. You mentioned that you were a mom, and I know you are. So what do you do in your house? What are some of your best practices for having a healthy home and trying to reduce exposure to these things?
So I think I have a two-part answer to this as well. And I think that I'm an environmental health scientist, an environmental epidemiologist. I truly believe that we can address and understand the impact of environmental health on children's development, and that we can use this data to make a difference. But one of the most important things I think that I've done in my work is working on a study where we looked at the interaction between the quality of the home environment and pesticide exposure on infants' development. And what we saw was that using some of our biological markers of exposures, we could understand that pesticides, when I was doing the study, we were focused on pesticides. Pesticides were associated, or we could predict about 6% of a child's IQ, or 6 points of a child's IQ using the environmental measures. But the strongest predictor in the positive direction of children's development was parental nurturing in a quality home environment, a stimulating home environment. So a home environment where kids are listened to, where they're read books to, where they're nurtured and cuddled, that these are the most positive, these can have the most positive impact on development. So we can't all go out and move further away from the West Side Highway. We can't maybe change where the bus depot is located. We can advocate for that. There are a lot of things we can do on the tips that we'll share afterwards about, cleaning, wet mopping, wiping down our windowsills, having vigilant practices about washing our hands and washing our fruits and vegetables. Those are all things that we can do to reduce our environmental exposures. But understanding that there are things that we can do in our homes that can also improve and sometimes mitigate those adverse exposures are strong predictors. So I think that's a very positive note to end with, is that there are things that we can do even when it seems like some of these things are totally out of our control.
And that is actually a great way to end it, because we're just about at time. So I want to thank you so much, Dr. Horton. This was fantastic. I think I learned so much as well. For everybody online, please be sure to follow our social accounts, Sinai Exposomics, to get real-time updates. I know we didn't get to everyone's questions. We'll try to do so online. If everyone, before we close, if everyone can complete an evaluation survey, we'll put the link in the chat and email it to those who registered. I think we are putting that in the chat at some point. And I'd like to thank everyone once again for joining and taking part in this year's lunchtime chats. And thank you again, Megan. That was wonderful.
Thanks so much, Maya. Thanks everyone for joining.